Swayam Prabha. Digital India. Educated India.
Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And so far what we have discussed about the structure of prokaryotic cell as well as the structure of eukaryotic cell, and also what we have discussed about the different organelles what are present in the eukaryotic cell, and we have also discussed their functions in the cell. And today what we are going to discuss, we are going to discuss about how you can isolate these different organelles so that you can use them for downstream applications. In the case of prokaryotic cell as well as in the eukaryotic cell, you have different regions which could be important for recovery of the product. And in this context, let us discuss what are the regions are present in a prokaryotic cell for isolating the proteins. So, as you can see in a typical prokaryotic cell, what you have the capsule, and then below the capsule you have the cell wall, and then below the cell wall you have the plasma membrane, and inside you have the cytosol or the cytoplasm. So, in the case of prokaryotic cell, you have the two major places where you can actually use to extract the proteins. What are these places? You have the cytoplasm, and the other place what you can use also is the periplasmic space. So, what is the periplasmic space? The periplasmic space is the space which is present in between. So, within the cell wall you have the outer membrane and then you have the inner membrane. So, within the cytoplasm you have in between there is a space, and this space is called as the periplasmic space, and this space can be used for storage of different types of proteins. Now, you can see the relative distribution of the proteins in the different subcellular structures. So, what you have is in the periplasmic space you have the 129 proteins, and whereas the protein which are present in the outer membrane or the inner membrane, you have the 50 in the case of outer membrane, and you have 21 in the case of the inner membrane, and then the major fraction of the protein which is present in the cytoplasm, that is the 183 different proteins. So, this means you have the choice of overexpressing your protein, or you have the choice to isolate the protein from the two region. One is the cytoplasm, the other one is the periplasmic space. So, let us see what are the steps you required to isolate the periplasmic fractions. So, as I said, the periplasmic fraction is present within the cell wall, and in the cell wall you have the two layer, one is called outer membrane, the other one is called the inner membrane. So, within this cell wall there is a region which is called as the periplasmic space. So, if you give some kind of osmotic shock, the osmotic shock to the bacteria, what will happen is the protein which are present in this region will come off and can be used for the downstream applications. So, these can be done simply by following a similar protocol or simple protocol. In this protocol, what you have to do is first you harvest the bacterial cell from the by centrifugation at 3000 G for 20 minutes at 4 degree. Then once you got the and then you carefully remove the media as well as the supernatant, and with the help of the pipette, and once you have removed the supernatant, you can gently resuspend the pellet in 1 ml of tris sucrose EDTA buffer, which is called as the TSE buffer, using a loop or using a pipette. And once the cells are suspended, you incubate them for 30 minutes in ice. So, this TSE buffer contains sucrose, and the sucrose is going to and this also contains tris and EDTA. The sucrose actually gives the osmotic shock to the cell, and as a result what will happen is the material which are present in the periplasmic fractions comes out into the supernatant. Once they comes out into the supernatant in this 30 minutes, then what you do is transfer the cell in a microcentrifuge and then centrifuge at a very high speed at 16,000 G. Once you spin at 16,000 G, it will give you the supernatant as well as the pellet. So, it will give you the pellet, which is actually the bacterial pellet, which contains the bacterial cell and the cytosol and other things, and then you are going to get the supernatant, and this supernatant is going to contain the proteins which are present in the periplasmic fraction. Now, for the isolation of the proteins from the cytosolic fractions, what you can do is you can take the cells and then you sonicate, and by doing the sonications you could be able to achieve the breaking of the cells. Once you break the cells, what will happen is the cytosolic content will be come out into the supernatant, and then what you do is you do a centrifugation as it was done for the periplasmic fraction as well. So, then when you do the centrifugation at 16,000 G for 30 minutes at 4 degree, that actually will give you a pellet as well as the supernatant, just like it was been discussed for the periplasmic fractions. And then the supernatant will contain the proteins from the cytosol, and the pellet will contains the dead or the damaged bacterial cell, which you can discard. So, this is for the prokaryotic cells. Now, talking about the eukaryotic cell, as we discussed previously also, the eukaryotic cells are much more complicated compared to the eukaryotic cell, and they contains well defined membranous organelles, and most of these organelles are useful for both for the study in terms of if you want to study the function of a particular protein, as well as some of the organelles are also being used for protein productions. So, whether it is a animal cell or a plant cell, in both the cases you can use some of the dedicated organelles for producing the proteins. Let us see what are these organelles. So, in a typical eukaryotic cell, what you have is the places where you can actually get the proteins in larger fractions, or the organelles which are having the high importance in terms of the research as well as for product recovery. Those are called as, so there are 4 or 5 place organelles which are having the very high importance as far as the eukaryotic cell is concerned. What are these organelles? One is called mitochondria, plasma membrane, chloroplast and cytosol. So, chloroplast is actually the protein production machinery as in the case of plant, whereas in the case of animal cell, the cytosol is the site for the protein production. But if you know the isolation of these organelles, you could be able to isolate either these organelles, or you could be able to remove these contaminating organelles from the cytosol, and that actually will give you a very high purity in terms of extracting the compound, extracting your specific proteins. And that actually also helps in terms of the downstream processing and make the purification of these proteins simpler. Before getting into the details of how to fractionate the eukaryotic cell and how to use them, let us see what are the technical tools are available to you to achieve the organellar separations from the eukaryotic cells. So, most of these fractionation of eukaryotic cell is done by the centrifugations or by separating these organelles based on the some of the basic physical properties. And there are two physical properties which people are using. One is called density, the other one is called as the ability of these organelles or the molecular weight or the mass of these organelles. This mass of the organelle is indirectly related to the ability of these organelles for their sedimentation in a given buffer or the given microenvironment. So, the instrument what you can use for the extraction for the isolation of these organelle is known as the centrifuge. So, you have different varieties of centrifuges which are available for R&D as well as for industrial purposes. You have the microcentrifuge where you can use the smaller samples like the Eppendorf's. Then you have the centrifuge with the larger capacities. These centrifuges can be used for two purposes, one for pelleting down the bacteria which are being grown in the large cultures like 1 liter, 2 liters, and then also these can be used for culturing for removing the cells, removing the dead and damaged cells from the cell lysate, which means these centrifuges can be used for clarification of your sample as well. These centrifuges are available which are either the temperature stable, means you can use them at 4 degree, or you can use at room temperature, that is 37 or the 25 degree Celsius. And apart from that, you also have the ultracentrifuge. These ultracentrifuges can be used to spin the sample at a very high speed, such as the 1 lakh G or more than 1 lakh G. The advantage of these centrifuges are that this allow you to separate the organelles which are very light or which are having the coefficient. This is the typical rotor what people are using for the ultracentrifuge, and this is another centrifuge where you can spin the animal cells, and this is being used in most of the cell culture applications where you pelleted the cells and you do the subculturing of these cells using these kind of cell culture using these cell culture specific centrifuges. Apart from the centrifuge machines, you have the freedom of doing two different types of centrifugations. You have the differential centrifugations or the density gradient centrifugations. In the differential centrifugations, what you do is you run the sample at different speeds or different RCF values, and because of that, when you spin down, the molecule which is corresponding to that RCF value get pelleted down, and you will find that particular organelle or the substance in the pellet fraction, whereas the supernatant will contain all other molecules. Once you take the supernatant, you can spin at a slightly more high speed, and that is how you can be able to separate the molecule based on their sedimentation coefficient or the sedimentation rates. Whereas, in the case of density gradient centrifugations, what you do is you exploit the ability of, or you exploit the density of a particular solvent system or the buffer actually. So, what you do is you vary the density of the external buffer, and because of that what will happen is the biomolecule or the organelles are getting separated because they holds the varying density, and whenever they matches to their corresponding density, they form a band or the zone, and that is how you can develop the different zone within the density gradient certifications, and every zone you can actually isolate later on and can be used or can be represented by a particular organelle. So, let us discuss these two techniques, how these two techniques can be used to separate the different organelles. So, as I said, in the differential certification is based on the differences in the sedimentation rate of the biological properties of a different sizes, shape and the density. You can imagine a situation where you have the particles of different sizes, shape as well as the density, and when you do the differential certification, in one round of certification these particles are going to settle down in the pellet fraction, whereas the all the remaining sample will be present in the supernatant, and the subsequent fractions when you do this supernatant you will get this fraction in the pellet, and the third time you will get these fraction in the pellet, and that is why it is called as the differential certification, because in one round of certification you have isolated these molecule, the second round you have isolated these molecules, and the third round you have separated these three molecules, and that is how you can actually separate if the samples are suspended in a particular solvent system, the small size, medium size and large size particles. Let us take an example. Suppose you have the different substances and the different material of different sizes, different density. For example, you have a iron block of 100 kg, you have a stone of 30 kg, you have the another iron block of 10 kg, you have a stone of 10 kg, then you have a cotton of 8 kg and you have another iron block which is of 1 kg. So, you know that this cotton is going to have the lower density, or it is going to be the lowest density compared to all other samples. Next to that is the stone, which is going to be slightly higher density to the cotton. Then you have the iron, which is actually going to be the highest density. So, if you do the differential certification, and what you will see is that the 100 kg block is going to be settled down first, followed by the 10 kg iron block and followed by the 1 kg block. Okay. On to that, if you further doing that, then you will see is that 30 kg stone block will be pelleted down. Next to that you are going to have the pelleting of 10 kg. At the end what you are going to see is that the 8 kg cotton pellet is going to be settled down, which means it actually not only depends on the molecular weight or the density, it depends on the both. It depends on the size, it depends on the density and it depends on the actual weight of that particular object. So, you can actually exploit these properties. Now, you can see another example where we have the different types of biological molecules. You have the microsomes, you have the mitochondria, you have nuclei, you have viruses, you have soluble proteins, you have DNA, you have ribosomes, you have polysomes, glycogen, which is another biopolymer. And what you can see is that all these molecules have the different sedimentation coefficient, different density. So, if you spin down them, what you will see is all these biomolecules will separate to each other because they will settle down at different steps. So, first round this molecule is going to be settled down, in the second round the DNA will be settled down, and ultimately in the third round, because the soluble protein is going to be settled down. Let us take a real example. So, in the real example, suppose we are isolating the different organelles from the liver, and what you can see is you take a liver from an animal. Let's first step you do the homogenization, that will give you the different cells, and once you do the homogenization, that will break down the organ and it will give you the individual cells. If you do the further homogenization, the individual cells will be broken down and then they will give you a combination of organelles which will be present in your vessel, and these different organelles can be separated, because what you do is first you will spin down at a very low speed, that is the 600 G for 10 minutes, and that actually is going to give you only for the nucleus or the heavy particles. So, that will settle down the nucleus in the pellet fraction, and all other molecules will be in the supernatant. Once you take that supernatant and put it into another round of centrifugation, and then you centrifuge at 15,000 G for 15 minutes at 4 degree, then it will pellet down the another heavy particles. These are called mitochondria, lysosomes and peroxisomes, whereas the remaining organelles will be present in the supernatant. Once you take these molecules which are present in the supernatant and do another round of centrifugation at 1 lakh G, which you will do in ultracentrifuge for 1 hour, what you will see is now it has pelleted down the plasma membrane, ER fragments and the smaller vesicles such as the which are called the light membranes. And now if you take the supernatant and do another round of centrifugation, and now you do a centrifugation at 3 lakh G for 2 hours, which also you are going to do in the ultracentrifuge, what you are going to get? You are going to get the very small particles like ribosomes, you are going to get the viruses, and you are going to get the free proteins. And if you take this supernatant, you are going to get the cytosol. This cytosol will contains the proteins as well as the salt and all other the liquid material which is present in the typical cell. Now, take another example. Suppose you started with the muscle tissue, exactly the same way you are going to do homogenization. Then if you do a centrifugation for 1000 G for 10 minutes, in first rot you are going to get the nuclei as well as the cellular debris. Once you take the supernatant and do another round of centrifugation at 10,000 G for 10 minutes, that actually will give you the mitochondrial fraction, and the supernatant if you spin down at 1 lakh G, that will give you the microsomes as well as the cytosol. And if you take these crude microsomes and run it for the density gradient, you could be able to even isolate or further divide or purify many materials from these microsomal fragments. So, what we are doing actually is that we are doing a repeated centrifugation at progressively high speed will fractionate a very complex homogenate of cell into their individual component, which means the individual organelles. And in general, the smaller the organelles, the greater is the centrifugation forces required to sediment it. Which means if you are working with the lighter and lighter particles, just like as we have taken an example of the iron and stone and cotton, you might have seen that we are supposed the cotton got pelleted at the end, because cotton is having the very low density compared to the stone as well as the iron. So, the lighter the organelles, the larger will be your centrifugation speed. Because of these limitations, you can also exploit the densities of these particular organelles or different molecules which are present inside the cell. So, in a typical different types of cell which are present in the biological system, you have the microbial cell or the prokaryotic cells, you have the mammalian cells, you have the organelles which are present in the mammalian cells, then you have the proteins, DNA and RNA. And as you can see, all these molecules have the very different densities. For example, in the prokaryotes it is a density between 1 to 1.15, whereas the mammalian cells are slightly different from in density from the prokaryotic cells. Similarly, the organelles are very much different from the whole cell, and they are in the range of 1.1 to 1.6, whereas the proteins DNA and RNA are also having the different densities, which means since these biological samples are having the different densities, you can also use this particular property as well to exploit and purify to separate them in a density gradient centrifugation. So, in a density gradient centrifugation, what you are supposed to do is you make a density gradient media. What is mean by density gradient media? For example, in this case we have taken a sucrose as a material. So, what you do is you put the sucrose, different concentration of sucrose starting from 0% to 30%, and as a result what will happen is that all these media is going to be separated in different zone of different densities according to the concentration of sucrose present in each layer. And then what you do is you overlay your complex solution which is going to be separated. Now, you can see that I have used the three different types of beads. One is blue, another one is green, another one is cyan, and what these beads, and then you do a centrifugation. Once you do the centrifugations, these beads will move towards the liquid, and because this liquid is containing the media with the different densities, it will actually going to create a friction or it will oppose the entry of these molecules, and the place where these molecules will stop is the place where the opposing forces as well as the centrifugation forces are going to be equalized, and that place is actually the place where the density of the media is going to match with your sample. For example, in this case the blue got matched here, the green got matched here, and the yellow got matched here. This means the if you have the samples with the variable densities, you can be able to separate simply by centrifugation of these samples in the density gradient, and as you run it you could be able to make the better separation between the molecules. And once the separation is over, you could be able to isolate the individual components from this particular tube, and you can take out the individual bands from these tubes, and you can be able to purify these individual bands into the individual fractions, and that actually individual fraction is going to give you the pure organelles or the pure material from the cell. How to collect these material? You have the two options. So, you can imagine we have started like this, and we run it for 1,50,000 for 3 hours, and that has given you the different bands corresponding to the different types of fractions. For example, in this case this fraction belongs to the sarcoplasmic reticulum fractions, then this fraction belongs to the light sarcoplasmic fractions, this fraction belongs to the triads, and this fraction belongs to the plasma membrane or the surface membrane. Now, what you and at the bottom what you have is the cellular debris. So, what you have, you can actually use the different way of collecting these fractions. One of the way you can do is you can manually put the pipettes. So, these are the typical pipettes which you can use. As you can see, these are the different pipettes, and what you can do is you can put a pipette tip in the media and you can easily suck your band of interest. The other way is that you freeze these samples and then you cut these tube in different slices, and then individual slice you can take out and then isolate the material which is present in that particular individual band. So, this is what we have discussed so far. What we have discussed about the presence of different organelles in the case of eukaryotic cell, and we have also discussed about the different regions which are present in the prokaryotic samples. We have also discussed what are the instruments which are available for you to isolate these organelles from the eukaryotic as well as the prokaryotic cells, and then very briefly we have also discussed how to isolate the periplasmic fraction or the cytosolic fraction from the from the prokaryotes. And in detail we have discussed the different approaches what you can use to isolate the organelles from the eukaryotic cells, either you use the differential centrifugations or the density gradient centrifugations. Thank you.